Hey guys, so now we're going to go ahead and talk a little bit about basic energy systems. The first one we've got is going to be the ATP PCR systems, the adenosine triphosphate phosphocreatine, which is going to be one that lasts us for about 5 seconds overall. We then have our glycolytic system. So specifically, we're talking about anaerobic glycolysis. So that's going to be when we're breaking down carbohydrates without going into the citric acid cycle and the electron transport chain. And then we've got our wonderful aerobic glycolytic system. So that's going to be the oxidative system, where we're going to go through and take that pyruvate, convert it into acetyl-CoA, run it through the Krebs cycle, also known as the citric acid cycle, and then those products of the NADH and FADH2 are going to go on to the electron transport chain. Now, beta oxidation is going to be the other aerobic system that we have. So there's actually two aerobic systems, and that's going to be the one where we're breaking down fatty acids, just cleaving off 2 carbons at a time. So ATP is the major energy currency of the body. So we have an adenine here, we have a ribose, which is a 5-carbon sugar, and then we've got 3 phosphates bonded together. So that diphosphoanhydride bond is where we're going to be getting the energy from when we break it off to make ATP and ADP, diphosphate, and into AMP if we break this one, adenosine monophosphate. Now, depending on how far we're going, and these are maximal efforts for each of these, we're going to see which energy system is providing the majority of our energy production. So we're talking about maximal 6-second sprints. Most of our energy is coming from creatine phosphate. The next followed by is just the pure ATP we just have floating in the cells, and then a little bit of anaerobic glycolysis, where if we're now increasing that duration to 30 seconds, we can see how the vast majority of that energy is coming from anaerobic glycolysis, and then a little bit is coming from aerobic glycolysis. And then we're going to keep shifting this further and further to more aerobic until, notice we're at the 2-minute mark. When we're looking at the 1 hour, it's going to be mostly aerobic glycolysis and a little bit of beta oxidation. And we're talking about 4 hours to run a marathon. You're seeing something more like a 50-50 split. So ATP PCR, this is going to be the one that we are going to be running through the fastest. It's anaerobic, and we're going to literally convert 1 phosphate from a creatine onto an ADP to replenish it into ATP. So it's a 1-to-1. This happens in the cytoplasm of the cell, also known as in the muscle, referring to it as the sarcoplasm. And notice it's going to be pretty quick. 15 seconds is definitely a bit of an embellishment. We're really talking about more like around 5 on average. And the higher intensity, the faster we're going to go. So literally, thanks to one enzyme, creatine kinase, we're going to take that phosphate off of creatine, take that energy and apply that phosphate to an ADP to replenish an ATP. Okay, real quick, real easy. Now, in order to replenish this phosphocreatine, we're going to need to get it to go back into the mitochondria of the cell in order to regenerate it. So you can kind of see how we have this interconversion of breaking down, building up, and then using creatine kinase, obviously, as another means to go ahead and allow for this replenishment. So we're going to see when we're doing this maximal effort, initially, we're going to have a slight drop in the ATP, but we're really going to be freeing up that phosphate from creatine. And that's when now we're going to see this drop off. Notice it's just 14 seconds. So this is going 100% hard as you can, really tapping all those fibers maximally. And then we think about how type 2 fibers, and specifically our 2X, are going to utilize these fibers or utilize this energy even more so. Another example of the contributions of the energy systems as we're starting from going maximally for 2 minutes. So the first 30 seconds, the next 30, and then so on and so forth. We can see that shifting more into our aerobic system. So the next energy system we have is glycolysis. Glycolysis is going to be breaking down glucose in either anaerobic, not oxygen available, or aerobic, oxygen available situations. This is going to give us pyruvate, which in the anaerobic is going to be converted into lactate. Aerobic is going to go to acetyl-CoA and then into the citric acid cycle. Glycogenesis is going to be where we're essentially synthesizing glycogen. Now, this is going to be occurring inside of our muscles and our liver. So it's going to be how we store it for long-term. Glycogenolysis is how we break it down into glucose 1-phosphate. So it's going to be a glucose with a phosphate bounded to the first carbon. That's why the number 1 is there. And this is going to occur obviously in the muscles and in the liver when we need to free up some for energy. Now, 12 steps that we're going to take from an original glucose or from glycogen cells, break it all the way down into ATP. This is going to occur inside of the sarcoplasm, and it doesn't require oxygen. It's taking a 6-carbon sugar. So this is the chemical model for, or chemical makeup of glucose, which is the specific carbohydrate inside of this. And it's going to break it down into 2 individual 3-carbon pyruvic acids, also known as pyruvate. This can be converted into lactic acid if we don't have enough aerobic capacity. If we're starting off as glucose, this system is going to yield us 2 ATP. If we're starting off as glycogen, it's going to yield us 3 because we don't require the activation energy of the first step at glucokinase or hexokinase, specifically when we're inside the muscle. So you can see how there's that activation energy that's going to require, hence we're only getting 2 ATP as opposed to 3, whereas we go from here, we skip that step, and we're going to go ahead and able to move much faster on through the system. So we are going to have 2 energy requiring steps at the beginning, and then we're going to have 2 steps at the end where we're going to be able to take ATP away. But since we've broken into 2 molecules and we're running each of those twice, it gives us this net of 2 ATP if we're starting from glucose, 3 if we're going from glycogen. Now, there's a lot of stages of glycolysis. First step is going to be phosphorylating. Second step is going to be cleavage. Third step or third phase is going to be our energy production. Here's a number of links you guys can look at for different YouTube clips and otherwise that is going to help you with learning this. Now, I'm not going to insult your intelligence and read these slides to you, but the key thing is what we're doing is taking that glucose, adding phosphate to, the 6th carbon, okay? That's the enzyme that's doing it, and this is the energy requirement. Isomerize, meaning that we're modifying it. So we're changing the molecule in such a way, but not actually adding or removing any hydrogen, oxygen, carbon, etc. So that's why we're now have what's known as the fructose 6-phosphate. So phosphate's still on the 6th carbon. Key is its move positions, and there happens to be our enzyme. Phosphofructokinase is sometimes referred to as PFK. This is a major rate limiting step. And this is now where we're going to add another phosphate, only now we're adding it to the 1st carbon. So once we go ahead and get it inside of the cell, we're going to phosphorylate immediately so it can't leave. And so it's either going to be put back into glycogen for storage, or we're going to go ahead and break it down and utilize it for energy. So this first phase is going to give us a requirement of 2 energy. And remember, PFK is going to be the major rate limiter. Now, phase 2 is literally just cleaving it in half. So we're going to cleave that fructose 1,6-bisphosphate into a dihydroxyacetone phosphate and a glyceraldehyde 3-phosphate. And then this is being the enzyme that's cleaving it is aldolase. Then that DHAP is going to be modified into another G3P, and that's through the triose phosphate isomerase. So at the end of that, we have a grand total now of 3 G3Ps, or sorry, 2 G3Ps to use numbers. Now, we're going to be converting this accordingly into what's known as 1,3-diphosphoglycerate. Okay, this is going to be producing what's known as NADH, and this is going to be important in the electron transport chain. So it's going to occur a couple times. So we're actually adding another phosphate. So notice that's why we're going to the 1,3. And in turn, this is going to be converted into 3-phosphoglycerate. So when we do so, we're going to be able to generate an ATP. And remember, we're doing this twice because we cleaved it in half. From there, we are going to now convert it into 2-phosphoglycerate. And in turn, this is going to be converted into phosphophenol pyruvate, PEP, through the enzyme known as enolase. Finally, we now are going to have pyruvate from that PEP, thanks to pyruvate kinase, which is going to give us 4 total ATP, because remember, we do each of those steps twice, and then 2 NADH, because we do each of those steps twice, where we only generated 1. Now, where it's going is going to depend on, do we have oxygen available? Do we have oxygen that's not available? Oxygen is not available, definitely going to be creating lactate through the enzyme lactate dehydrogenase, which immediately dissociates into lactate and a proton. Lactate is a carbohydrate. It's a 3-carbon sugar. It's useful for energy. The proton is what gets you, because that's what drops your pH, makes it pretty uncomfortable to try to keep up doing whatever you're doing. So is lactate the problem? No, it's the proton that drops your pH, because the pH gets to be too low. Literally, the enzymes of glycolysis are not going to work. Is the system trainable? Yes, we can increase the number of these enzymes, as well as we can enhance our ability to utilize lactate as a fuel source from the skeletal muscle that's type 1 adjacent, along with the fibers throughout the rest of the body. So we're not going to have as much of accumulation of lactate, which really it's not the accumulation of lactate, it's the accumulation of protons that drop our pH that causes performance to suffer. So We produce a total of 4 ATP, but we only net 2 because of the 2 we use for activation. We're only using carbohydrates in glycolysis, specifically glucose, and breaks down the 6-carbon sugar into 3 carbon structures, lactate and pyruvate. Now, our oxidative systems are going to be a different beast altogether. This requires oxygen in order to produce ATP, and this all occurs in the mitochondria inside of our cells. This gives us way greater amount of energy yields. However, it requires obviously oxygen, which is much harder to get enough to whenever we happen to be doing very high intensity events. So this is something where we're talking about still moderate to high intensities, but we're talking about endurance, so sustained efforts. So we oxidize the carbohydrates to get that pyruvate, which is going to be converted to acetyl-CoA, and that's what's going to feed into the Krebs cycle. And then all the NADH and FADH2 is in turn going to be fed into the electron transport chain. Now, beta oxidation is cutting off 2 carbons at a time off of however long that fatty acid is. Each time that gives acetyl-CoA, powers in, goes on. So mitochondria itself, you guys can see that being A. B are the little glycogen granules, and C is actually going to be an intramuscular lipid source. So we can see the mitochondria under even more intense electron microscopy. You can see the matrix space, you can see the outer and inner membranes, and the intermembrane space, which is where we're going to find that wonderful gradient that's being used by the electron transport chain to generate energy that in turn that ATP is going to migrate outside. So we can see the number of mitochondria you're going to have in those skeletal muscle fibers, as well as the basics of that pyruvate. In turn, we're going to remove a carbon, so now we're down to a 2-carbon molecule. We're going to go ahead and get ourselves an NADH, and then it's going to be bound up to coenzyme A, hence acetyl coenzyme A, which in turn removes the CoA, and that acetyl group goes through the citric acid cycle. Yay! And there's the enzyme metabolysis. Now, here are all of the steps of the citric acid cycle. There's a lot going on. I don't fully expect you guys to memorize every single part of it, but it is important that you understand it and you understand how it flows from one step to the other. So how we feed in, how we're going to move forward, how notice we're adding water, and we're removing carbon dioxide and developing both NADH, FADH2, and specifically, when at succinyl-CoA synthase, we're going to be actually generating a GTP, which in turn is going to convert immediately into ATP and be utilized for, obviously, energy inside of the cell. So glycolysis, remember, is in the sarcoplasm, Krebs cycle, electron transport chain in the mitochondria. And this is where we're going to be able to undergo this metabolic effect. So if we're thinking about the muscle cell itself, glucose comes in, gets phosphorylated in G6P. That in turn is going to go through glycolysis to give us pyruvate. Pyruvate in turn is going to go into the mitochondria, be converted to acetyl-CoA, and then undergo the citric acid cycle. Now, what we're going to find is for every single NADH that we're generating, so we're getting obviously a grand total of 1, 2, 3, 4 of them from the citric acid cycle, or sorry, from glycolysis, and then we're getting a number of them as well from the Krebs cycle. So this is why we get 6 of these, because we're going to get 3 each time and running it twice. We got 1 FADH2, but running it twice. We're going to be getting about 3 ATP for every NADH and about 2 ATP from every FADH2. The numbers can be a little different when we work it out later. So the electron transport chain is coupled with it, meaning they're both going to be occurring at the same time. And we're creating this major hydrogen ion gradient effectively from the mitochondrial matrix to the intermembrane space, which in turn is going to be moving what's called complex 5 later on. And that's going to be where we're producing energy. So NADH and FADH2 are going to do a number of things. First and foremost, they're not going to allow development of those protons. So we're not going to be able to as greatly acidify both muscle and blood. It's going to carry this into the electron transport chain. So it's going to be that intermembrane space where we're going to use it to develop that gradient. And then when we utilize oxygen with it, we're actually going to form water. So the major essential products at the end of all metabolism of food aerobically is going to be carbon dioxide and water. So that NADH is going to free up its energy, which is going to pump protons into that intermembrane space from complex 1 through complex 2 through complex 3, and then on through complex 4. FADH, on the other hand, is going to feed in through complex 3. So you can see how complex 2, we've got that FADH where we're going to notice succinate dehydrogenase is going to be the exact same location And in turn, this is going to move these 2 electrons, which are going to essentially power this pump. So there's no transporting of protons here, but instead that energy is moving on to the further components in the actual complexes. So complex 1, NADH, complex 2, FADH, and in turn, this is going to go through CoQ, which in turn is going to pump more protons through complex 3. If you need me to pronounce these names for you later, guys, it's more than fine, just let me know. Then cytochrome C, in turn, is going to influence complex 4 to create that gradient even more so. And then we're going to combine that at complex 5, which is baller because it looks like a freaking revolver. This is literally the final step of the electron transport chain. And like I said, guys, it actually looks like a freaking revolver. So it's mounted to the space. It's going to rotate thanks to this proton pump, and the alpha and beta folding of the proteins in turn are going to take the 3 chambers of this revolver and it'll twist. And as it's doing so, it's going to go ahead and create these ATP from taking ADP and a phosphate binding together and shipping on. So it's actually going to keep rotating through. So you can actually see the microscopy down here of how it's going to move from one configuration to the next, and that force is literally going to make that go. So at the end, we're left with carbon dioxide and water. That's it. But when we tally together all the ATP and the NADH from glycolysis, converting pyruvate into acetyl-CoA, and then from the citric acid cycle together, we see how we get this massive gain of energy. And so how much we're going to get is not going to be that pure 3 and 2 number you saw earlier. It's more like 2.5 and 1.5. That's because we have to use a certain amount of energy in order to get that ATP back into the sarcoplasm, as well as, notice guys, it's going to be a total of 10 pumped through, but we're get 1 ATP for 4 NADH and 6 for FADH2. So when we go ahead and actually break that down fully, the amount that we're going to get is actually going to be a little bit lower than what one would expect. But it turns out it's still way more than the 2 to at best 3 we were going to net from anaerobic glycolysis. So, yay. All the energy producing. Now, when it comes to fat, we've got just pure fat cells, but here's that lipid droplet next to the mitochondria and then the muscle fibers themselves. Now, this is great because notice it's right next to the mitochondria. In turn, we're going to find inside of the cell, we're going to activate those fatty acids. And now this is going from, notice inside the fat cells, taking the triglycerides, breaking them down through hormone-sensitive lipase into glycerol and 3 fatty acids, which will then transport through the bloodstream, typically bound to albumin. There's 5 high affinity sites. A little bit will float free, but very little. It will then transport into the cell, be activated, and then be transported into our mitochondria thanks to the CAT1, CAT2 transfer shuttle, which is literally using carnitine. That's part of the reason we see it in energy drinks, the idea that it's going to help you metabolize fat a little bit easier. And from there, we are going to undergo beta oxidation. We pull off 1 acetyl-CoA at a time, feed that into the Krebs cycle, and then those products in turn are going to go into the electron transport chain, and then finally with complex 5 generate ATP. It's a lot. Listen to it multiple times if you guys need to, so you understand it. So break down triglycerides, so 3 fatty acids bound up to 1 glycerol into those basic components, put it into the bloodstream, where in turn, it's going to be broken down into acetic acid, converts to acetyl-CoA. That in turn is going to go through the citric acid cycle and electron transport chain, and it requires way more oxygen to get this party going. However, it generates a lot more energy. So beta oxidation is really just a 4-step process that occurs for every 2 carbons that are getting cut off of the fatty acid. So you can see how we got that fatty acid. We're now going to go ahead and create it into an unsaturated fatty acid, which in turn is going to now get into essentially a different modification and then eventually a keto acid. So we're generating NADH, breaking off the 1 acetyl-CoA, and then generating an FADH2. So it literally is just going to keep chopping this long fatty acid down 1 at a time. So the carnitine gets it in. Once we're in there, we're just going to chop off 2 carbons, 2 carbons, 2 carbons, 2 carbons, and so on and so forth. And each time we do so, we generate an NADH and an FADH2, which obviously are going to both give us that 2.5 and 1.5 respectively ATP generated per. So what we're going to find, as long as we have oxygen occurring, we're going to go ahead and get the same products we did before of ATP, carbon dioxide, and water. Now, we can get way way more ATP when we're breaking down free fatty acids than we can with carbohydrates. So getting back, thinking about all those steps, if we have an 18 carbon fatty acid, we're going to be able to go through this process with acetyl-CoA 9 times, and we're going to go through beta oxidation 8 times because you only need to chop in those segments as many times as you have those chains, -1. So altogether, you can see how just from palmitate or palmitic acid, you're going to have 16 carbons, which in turn means you're going to be able to cut that, notice, into 8 2-carbon molecules, and you'll have to do it 7 times. So hence this generation. So altogether, your ATP, how much you're deriving, is massive per unit of size compared to fatty acids. And so that's why typically at pretty low intensities, we're getting the vast majority of our energy production simply from utilizing fatty acids as energy, which is a good thing. This means we're going to be able to preserve our glycogen because we're not going to store it anywhere near as much as we are with our fatty acids. So our ATP PCR system and anaerobic glycolytic systems don't give us a whole lot of energy, but they are going to allow us to get that energy very rapidly, especially when we're doing super high intensity exercise. The aerobic systems are going to give us way, way more energy, but it's going to deliver it slowly. And that's going to be limited by, can we deliver enough oxygen to those tissues? So the amount of ATP we're going to get is going to be pretty solid when we're thinking about getting just 1, just 2, and now we're talking about in the 30s. And then we compare that to fatty acids, we're going to get way, way more. Now, protein is going to be giving us maybe 5 to as much as maybe 10% of our energy demands when we're playing sports, being active, but it's moving at a much slower rate. And obviously different fiber types are going to be much better at utilizing different types of fuel sources, meaning because of the aerobic capacities of type 1 fibers, the amount of capillaries, the amount of mitochondria, they're going to be very good at utilizing fats for fuel. Whereas things like your type 2 fibers, not a lot of capillaries, not a lot of mitochondria, they're going to use preferentially anaerobic glycolysis and ATP PCR. So thanks for listening, guys. Stay safe out there. Bring questions on this, and we will talk all about this next week. Bye-bye.